much for showing up, to whoever you are, wherever you are, and whenever you are listening to this talk. Again, my name is Anthony. I've been with the team since the beginning, and over the past 5 years, Rapid Novor has really specialized and perfected the craft of monoclonal antibody protein sequencing using mass spec. And recently, as some of you are probably already aware, we've been talking about polyclonal antibody sequencing as well. So over the past several years, we've been able to work with many of the major pharmaceutical companies across the world. We've been able to help many major academic institutions such as MIT, Harvard, UCSF, Oxford, and Cambridge in the UK. And we've sequenced over 2,700 different monoclonal antibodies or protein using our mass spec-based approach. We've really perfected the art of, tearing down a protein of interest, pulling out mass spec data, and reassembling those spectra into full-length antibody and protein sequences using our proprietary Novor and REMAB sequencing technologies. So what we're going to do today is walk a little bit through different applications in early stage research and development, which is usually where a lot of our clients are coming to us and working with us. There's been a lot of interest in working with us from teams in therapeutic antibody development, teams that are keen on characterizing an immune response that could be in response to an immunization protocol, in response to a vaccine, or something like that. Indeed, we've also worked with many teams for biomarker discovery and diagnostic development, and of keen interest to some of you here today is the idea of converting a PAB to a MAB or a MAB cocktail. And of course, we'll be going through all these different cases throughout the presentation. But I thought it might be relevant to start off by looking at some of the other approaches in conventional antibody discovery and certainly in antibody sequencing. So obviously, you're all probably familiar with these technologies more than I am. We have standard kind of hybridoma methods of antibody discovery. We've been working with combinatorial approaches such as display libraries like phage and yeast display. And of course, people are consistently using approaches like B-cell cloning as well, taking a patient of interest, sequencing those individual B cells, either or doing like a FACS sorting kind of thing and cloning those monoclonal antibody sequences and then using those for their purpose of interest. But of course, you could go about discovering new antibodies, but Rapid Novor has really specialized in the art of resurrecting or saving old antibodies that have already been discovered. Many clients have come to us actually because they've got an antibody in hand that works and is fit for purpose and does the job of binding against a target of interest in their particular configuration quite well. But the problem actually might be that they can't test it or use that antibody anymore for one reason or another. And so they'll come to us and say, Anthony or Rapid Novor, I've got a little bit of this antibody left. I would like to try to capture that antibody's amino acid sequence so that I can create a recombinant. And indeed, this is where we see a lot of interest, and we've been able to help so many different clients over the past several years. This is the workflow in general. We're going to go in a little bit more detail here over the next several slides. So although that overview kind of runs very quickly through it, we're going to take a bit more of a nuanced approach as we kind of go through the process. So we start off with enzymatic digestions. We can then load those digests into the LC-MS system and acquire mass spectra. We can then perform the REMAB peptide sequencing and then a subsequent assembly into the full-length antibody or protein of interest. And then, of course, our expert bioinformaticians will review that data and ensure 100% accuracy in terms of the protein of interest. So it all starts usually with a gel. We like to see how pure the protein of interest is. This is particularly relevant in the context of a mass spec because we are able to really sequence the dominant antibody of interest in the sample. This is relevant as we go further along in the conversation, particularly when we talk about polyclonals as well. But this is one of the reasons why we've been able to make our name in the monoclonal antibody sequencing business. We've been able to take these purified monoclonal antibodies and pull out their amino acid sequence in the absence of other protein species. The gel on the left, excuse me, take a sip of water. The gel on the left, as you can see, is a nice antibody. We can see the heavy and light chains separating quite well, and of course, they resolve nicely on the mass spec. The gel on the right, however, if you're unfamiliar with this kind of gel, this is a lot of BSA next to a couple of really good antibodies. Luckily for us, though, we've been able to work with these samples quite well. We've developed a robust protocol for handling these kinds of samples. We usually do a protein AG purification scheme in which we can remove everything other than the IgGs, and at the end of that, what we're left with is just the IgGs, hopefully of the antibody of interest. But of course, if there's a lot of BSA in the sample, it's often the case that we'll still see a lot of mouse antibodies, even in the purified sample. But that's not a problem. We've luckily, over the past, like I said, several years, because we've sequenced so many, our team has practically memorized the sequence for BSA and many mouse proteins, and as a result, we're able to usually work through those projects as long as the dominant antibody is the antibody of interest. So, in a kind of for those of us that are visual learners, a pictorial example serves us well. Simply state, like, once we receive the antibody sample of interest, a small aliquot, usually about 100 micrograms, we can digest it into different peptides. Different peptide colors, as you see, denote the different enzymes that we use in order to digest that sample. We then have those digests put on the mass spec. The mass spec then gives us spectra, and then we can relate that data and convert it into peptide sequences. Those peptide sequences can then be assembled into a full-length contiguous protein sequence. And in the next slide, we see what our data actually looks like. So for those of you that are unfamiliar with mass spec-based sequencing approaches, I'll quickly walk you through one of these examples. So at the very top, we see the bar that denotes the area of the antibody. The top bar, you see green, orange, and blue. The green denotes the variable regions framework. The orange is your CDRs. And then the blue is your constant region. Of course, this is an excellent case. This is NIST, and so it should look good, but it does, which is reassuring because in terms of like how we benchmark ourselves, when I started with the company, we were hoping to achieve at least 3 to 5 overlapping peptides of coverage over every single amino acid, including the CDRs. And if you look in the literature, I think that would be a really high score to achieve. Certainly, if you investigate our contemporaries in an academic setting. However, experience serves us well, and so we've noted that we always strive for the higher levels of coverage and thus higher levels of accuracy in terms of recapitulating the exact amino acid sequence. We've been working on this problem for many years. Obviously, one of the end goals was to achieve 100% accuracy in sequencing monoclonals, but we've always been working on this problem of polyclonal antibody sequencing in the back of our minds. And luckily, I can say that with this level of coverage that we've achieved in this kind of pure monoclonal antibody case, it serves us well as we move forward into the polyclonal antibody sequencing technology, which I'll be able to show you shortly as well. So, of course, some of you might still be asking, like, why are we doing this? Mass spec's expensive, it takes a long time, I'm not sure if it's completely accurate or not, and indeed those are fair questions and that need to be answered. But I'm here to tell you that there really is a legitimate interest in this, as expressed by not only the fact that we've literally sequenced thousands of individual monoclonal antibodies, and we've worked with many of the major pharmaceutical companies and many different clients worldwide. But of course, like there's real interest in that, and they come down to usually these main topics. So, firstly, if you have the hybridoma and it's producing your antibody just fine and everything coming along, then obviously you're probably in a good case. You don't necessarily need to work with us. But what if that isn't the situation? Like, what if you're working with a hybridoma and you've been using an antibody from it for a long time, but maybe now that hybridoma is a little bit less stable, it's producing a different antibody, or now you're dealing with issues with respect to the binding affinity of that antibody, or mutations that are present in the original myeloma cell line. These all can be contributing factors that could lead to a genetic drift in that original cell line, and thus a difference in terms of the expressed antibody that you're trying to work with. It might not even be you, it might be someone else that's selling you that antibody that's having that issue, but nonetheless, fundamentally, there's a problem with the antibody of interest that you've been working with. And that could yield a lot of issues, such that like you only have that physical protein left to work with, and you might be on a limited supply. As Bradbury has noted a couple of years ago, and others, there could be this other issue of additional light chains or additional heavy chains present in the sample. There could be a number of different reasons that lead to that event. Most common for us has been the presence of an additional light chain, which is an artifact from the fusion of the myeloma cell and the original B cell used in the hybridoma development. The myeloma cell line could actually produce an additional light chain. We'll see it in our samples pretty regularly, and indeed that can be an issue when you're trying to work with that original antibody. If you don't know that there's an additional light chain present, that could lead to issues with respect to affinity as a result of a loss of avidity, because you have this kind of bispecific antibody being formed, and of course that's problematic in terms of just like the straight affinity of the antibody of interest. But further than that, if you actually decide to go into the hybridoma and try to sequence it, you could actually see the wrong light chain present in your sequencing data. And of course, if you try to create a recombinant and express that antibody, you're going to find that the recombinant antibody that you derived from that genetic sequencing data is actually wrong as well. And thus you say, oh, well, what do we do now? Those are usually the best options to exercise first, but like in the absence of those solutions, you might want to consider something like protein sequencing as well to capture that antibody. Because it's not just about the cost of developing a new antibody or finding a new antibody, right? Like some of you might actually have built an entire assay around an antibody of interest, and that could cost a lot of money for a lot of different reasons, right? In terms of validation, in terms of actually paying someone to develop that antibody-based assay. Anecdotally, I've heard teams spend hundreds of thousands of dollars in terms of developing an antibody-based assay for their application of interest, only to find that they've lost access to that antibody. You could develop another antibody for a lot less than that, but obviously developing the entire assay, again, around an entirely new antibody is another matter entirely, right? So aside from that, we've seen issues where people can't source an antibody. Maybe you're working with a vendor. Maybe they've been selling you an antibody for the past couple of years and you've been running preclinical assays with that antibody of interest. But now, 2 or 3 years later, they run into one of the issues I mentioned above. Now the antibody that they're sending you is different, and thus you've got a different antibody in your hands and on your preclinical assays, and you're heading into, submitting that data for evaluation, and you can't recapitulate the original results that you've been working on, developing for the past couple of years. And that's a big problem, right? And in those cases, again, you can just send us whatever you have in terms of the physical antibody protein. We can sequence the amino acids. We can give you the ability to create a recombinant antibody from there. And lastly, I've got a picture of Twinkies here. It's kind of tongue-in-cheek, but it's happened to others before, where the supply chain just becomes limited. So maybe, like for the reasons I just mentioned, you want to preemptively ensure that you have indefinite access to your antibody of interest as a result of these kinds of problems that can pop up unexpectedly in your hands. So, okay, I'm done talking about monoclonals for now. For those of you that are still sticking around, you might be keen to learn a little bit more about the polyclonal antibody sequencing technology that we've alluded to in the title and in the description of the chat. So this is the workflow. Those of you who have never seen it before, essentially what we do is we start off with an animal of interest or patient of interest. We pull down the blood, and then we can perform some specific purifications of the serum as well as the PBMCs, allowing us to create a proteogenomics database and yield full-length antibody sequences at the end of that that are paired. In a little bit more of a granular approach, we have 4 phases to these projects. We have an immunization step, we have a screening step, we have a deep sequencing, combinatorial database, combination of databases step, excuse me, and in addition to a synthesis step to validate those antibodies of interest. In more detail, of course, we start off with immunization. We always like to get some pre-immunization bleed, a naive bleed if we can help it, a test bleed as well as a final bleed to assess for titers, but preferably also to actually use some of that blood in our sequencing analysis. We like to do a protein AG purification to pull down only the IgGs in the serum, and then subsequently perform an antigen-specific affinity purification so that we're only going to be pulling out binders against your target of interest and negating it and leaving everything else washed out. Of course, we're going to do an ELISA screening for titers, so hopefully we're getting high titer rabbits or patients. But of course, the real test is going to be in phase 2. So phase 2, we've really developed and built and changed over the past several months since we started doing this to mitigate risk and improve the probability of success in downstream efforts. So in this screening, we're able to assess up to 5 different samples. So that could be 5 different rabbits, that could be a couple of different fractions against like a different peptide immunization protocol, but essentially it's a flat fee and we offer up to 5 different samples to be sequenced, to be screened. But effectively what we're doing in the screening is actually trying to sequence them and seeing what kind of data we get back. So this is where we do what I described earlier, the multi-enzyme digest, the bottom-up mass spec, and true de novo protein sequencing. Other people have tried doing proteogenomics as well in the past, but one thing that we like to kind of just point out with respect to our assessment is that we're doing true de novo protein sequencing throughout our process. Of course, we do reference a genomic database, but ultimately the data does stand on its own 2 feet with respect to being a de novo protein sequencing analysis. And it's only if this phase 2 screening is positive for a sample of interest that we will allow it to proceed and move into phase 3, where the probability of success is is significantly higher. And in phase 3, this is where we do more of the same. So we'll do an RNA-seq of the IgG amplified RNA from the PBMC sample prep. We're going to combine the genomics and the proteomics databases, and then do the complete de novo sequencing of the most abundant antigen-specific IgG high, heavy, and light chain constructs. And in addition to that, once we have these extremely well-sequenced heavy and light chain constructs, we can actually go back and do some other, use some other mass spec techniques to do pairing of those heavy and light chain constructs into their cognate ligand. Of course, we need to validate the antibodies that we sequence, and this is how we can do this. So we do actually recombinant antibody expression, pretty straightforward. We synthesize the gene of interest, transient transfection in the HEC293 cells. We express those IgGs, and then we ship them off for validation by the end user. So that's the idea. How did we do? Well, to address one of the immediate questions that many of you have is, can you convert a PAB legitimately into a MAB or MAB cocktail? Happy to say the answer is yes, we can and have done it. We did do this project actually already several months ago, and we have many projects ongoing this year as well. Some are in COVID, some are not, but we are indeed able to and capable of and have successfully done this protocol where we, in this case, the data I'm about to show you was against a peptide antigen. So we immunized rabbits with this peptide of interest, pulled out the polysera, put it through the workflow I just showed you, and here were the results. So as you can see, the data looks actually quite strong, despite the fact that this is a polyclonal antibody and there's a lot of different antibodies inside of the sample. Our data looks really good. A benchmark for us with respect to monoclonal antibody projects has always, over the past year or so, has been to get at least 20 to 30 peptides overlapping over every amino acid. And indeed, in this case, we did achieve that level of coverage as well. As you can see, the data looks strong. in this antibody 1, and similarly in this antibody 3. As you can see, if you look at the heavy chain and the third CDR, heavy chain CDR3, the data looks very strong. It's always a reassuring fact. As long as we're nailing CDR3, we feel very confident about those results as well. In fact, we felt so confident that we decided to express those antibodies, and here are the results by ELISA. As you can see, R1 and R3 faithfully recapitulated the binding activity of PD030, which was the original polysera, and we felt very good about this. We did include some negative controls as well. R4 and the strict negative control were, for all intents and purposes, a negative control. R2 we did express as well. It didn't turn out to bind as well as the R1 and R3, but nonetheless, as you see, we did express 2 antibodies that faithfully recapitulated the binding activity of that polysera, and they work quite well. They work quite well even to this day. These are antibodies that we developed for internal applications, and in fact, they do really well. You know, a year later after we've completed that project. So, of course, some of you might be curious now, what do we need to get started in case we want to go forward with this? So that's a great question, and I'll say this. Pretty much we need 2 pieces of starting materials. We need the actual IgGs that you're interested in sequencing from the poly serum, and we preferably also will want to have the blood. In fact, this is a requirement, a strict requirement for us. So I'll start with the left, and then I'll go to the right-hand side. This is a pretty easy way to interpret the data here. It's like a go, no-go. Let's say you have a target in mind. Great, we can work with you. We obviously work with rabbits and patients that we can acquire samples for. So if you have a particular target of interest, we can work with you to design you know, a workflow that works for everyone with respect to your specific project. Let's say you have the rabbit or the patient's blood and you have the affinity purified material. Perfect, great. We don't need to do any immunization or find those samples. We can just work with what you already have and proceed from there. Let's say you have the rabbit or the human blood, you have the affinity purified material, but you want to do the NGS work, or you already have the data, like great, no problem. We can work with you at that stage as well. But a no-go is a situation where like you just have the affinity purified material. Unfortunately, at this time, we can't faithfully put the workflow through without having access to the B-cell repertoire accompanying the immunization in the organism of interest. Let's say you just have a polyclonal antibody that you've been working with. Again, sorry to say that unfortunately there's just no way for us to currently perform like a protein sequencing only approach and deconvolute the most abundant antibody species in a polyclonal antibody sample. I know it's of particular interest. I know many people are keen to see this solution presented and offered as a service. I'm keen to work with you, trust me, but it's just not available at this time. Hopefully, in the coming months and years, you know, we'll have something more to offer to that end. But sit tight. You know, you can always subscribe to our mailing list. You can always visit our website. Check us out on Twitter, Instagram, LinkedIn, whatever. You know, we're always going to be posting important news, particularly that piece of news. I know everyone is excited to see that come to fruition. Okay, let's say you're working with other animals other than rabbits and humans. Let's say you have like a goat, sheep, llama, mouse, hamster, whatever project in mind. Unfortunately, at this time, we just aren't able to work with those different species. I will point it out a little bit later on, but that's something that we're working on as well. We are aware of the need to work with polyclonal antibodies that are produced in these other organisms as well, and we're keen to develop solutions for them. It's just that they're not available quite yet, but they will be in the coming years, coming months and years. It's nice to point out that we recently actually acquired some new talent to develop more of our NGS pipeline internally as well. So, particular applications in mind. Of course, some of you are sitting here today looking at your therapeutic antibody development programs and wondering if this can plug in. I would hope that it can, and I'll kind of talk about that here. So, of course, many of you have seen this kind of thing before. The notion that you're starting off with, you know, thousands of different antibodies that might bind against your target of interest. and you're going to whittle that down through your characterization assays and coming down at the tail end with like a number of different, a much more strictly selected number of candidate leads to proceed in your therapeutic antibody development program. Of course, I'm happy to tell you that we'd be keen to explore those possibilities as well. Finding neutralizing antibodies against tough targets is an area of keen interest for our team and our technology. We've been working with COVID-related projects, as most people have, throughout 2020 and 2021, and indeed those results are forthcoming using our approach, where we did actually work with a number of convalescent patients to sequence monoclonal antibodies present in those patients that are highly neutralizing antibodies. And of course, we believe that we could apply that same workflow against other diseases of interest and yield similar results. Of course, coming back to the point I made earlier about antibodies of interest, with respect to monoclonal antibodies of interest, it could be the case that, if you look on the right-hand side, that assays are your game. You're working with PK/PD assays, you're trying to quantify particular metrics of interest, and it might be the case that you're just looking to develop a monoclonal antibody if you can't already find one. Indeed, this is another area in which we've been able to find quite a bit of interest. And coming back to the point about therapeutic leads, the sequence optimization aspect of this, this is an argument. This isn't, let's say, like a demonstrably proven fact about, human antibodies. But of course, the theory goes that developability profiles are better with human antibodies, and indeed, sequencing monoclonal antibodies directly from patients of interest might yield antibodies that actually have better developability profiles than a hybridoma-derived or phage display-derived antibody of interest. Although I know that others might disagree with that point. This is our conjecture, this is our point, this is our bias, because this is, the name of our business. But indeed, yeah, this is where we've seen some interest. So of course, antibody validation could be of keen interest here as well in your therapeutic development pipeline. As I mentioned earlier, you might be building an assay. You might be building an assay against a target, and you're trying to validate, your antibody for specificity, for high affinity and things like this. Let's say you started off with 10 or 15 different commercially available antibodies, and you've been working with those and developing your pipeline, but actually none of them really worked out all that well as compared to a molecule that you might have in your hands already. In those kinds of cases, again, remab might be the way to go. It might be the best strategy for you to just take an antibody you already have access to, sequence it, create a recombinant, and ensure indefinite access to it. Pardon me. Stepping back here. Okay, we should be on this side. Okay, cool. And similarly for the antibody development and discovery aspects of your workflow, repab might be the alternative option here. Discovering an antibody against a tough target, a domain-specific target might be of interest to you in your therapeutic antibody development. Also, good to know is that we've got friends in the industry. So we've been working with Absolute Antibody for quite a number of years now, and they do what we don't, which is antibody engineering and recombinant expression. So they're able to actually take your antibody of interest, reformat it, recapitulate it into a recombinant antibody, but they might do things like switch the subtype. So changing your IgG4 into an IgG1 or IgG2 or whatever it is, making it a CFE, bispecific, maybe converting a rabbit antibody into a different species of interest. You might be keen to see that rabbit antibody plugged into a human backbone with similar CDRs. Of course, this is what our partners can and have done for a number of our clients over the past several years. And of course, the other applications that we've seen a lot of interest in have been in characterizing the immune response, but also specifically biomarker discovery for diagnostic development. Of course, there's a lot of different ways that you can do diagnostic projects. Of particular note, we've seen some interest in ADAs. I think that this particular assay was built around antibodies that probably needed a higher level of specificity, and they were finding higher levels of specificity using rabbit antibodies than the mouse antibodies that they had generated. I know there was a lot of debate about mouse versus rabbit and things like this, but of course we found that some of our rabbit antibodies are extremely specific to our targets of interest. They don't bind against off-target, high homology targets that are present in the sample as well. And luckily, like there's enough, rabbits are big enough for us to actually do our workflow on because we do require quite a bit of material. Other applications have been in immune precipitation. So certainly this is a robust method, relatively easy to set up if you've already got a good antibody in your hands. But of course, that therein lies the problem, right? Like it could be the case that you really require an antibody that's fit for purpose and you're having difficulty accessing such an antibody. Depending on the antibody of interest, you could have issues with homologous antigens, and there could be a legitimate problem with signal-to-noise ratio with respect to using an optical method to assess an antigen of interest using your antibodies. And so this is one of the reasons why we've been working on the Novor platform. The workflow works something like this, where a proposed workflow could work something like this, where you have a blood sample of interest, it's full of lots of stuff, but you really only are interested in your one antigen of interest. Using an antibody that you've acquired or we've discovered or whatever it is, using that antibody of interest, you can precipitate out just that antigen that binds to your antibody. And of course, we can then use mass spec to actually do a relative quantitation of that particular analyte. This is where the value of the technology really comes in. As compared to something like an optical-based method, we can strictly analyze a specific analyte of interest and then perform a relative quantitation over time. Luckily, we've actually developed a pretty robust platform for this, which is seeing some use in clinical cases where of particular interest to us was in immuno-oncology, where we have a particular indication where patients are producing a lot of a monoclonal antibody, and they'd like to track that monoclonal antibody over time. Because of our sequencing technology, we can actually offer a significant improvement to the current standard practice, which requires a highly invasive bone marrow aspiration. Our technology allows us to just simply extract a bit of blood, do de novo protein sequencing against that monoclonal antibody, and then subsequently track its relative abundance over time using a proteotypic peptide unique to that patient of interest. Similarly, like developing these kinds of assays using mass spec can yield high levels of sensitivity while not having to dig too deep into that patient of interest with respect to pulling out bone marrow or some kind of other genetic or some kind of other material which has the genetic components that you're looking to do a high sensitivity assay on. And of course, I'd be remiss if I didn't mention the stuff that we're working on. So Novor is, of course, as I mentioned earlier, developing custom NGS solutions, things like novel primer sets. One of the reasons why we work really well with human and rabbit samples is that they are big enough. We can actually extract enough material out of those organisms in order to actually run the proteomics pipeline. But also, from the NGS perspective, there are actual primer sets available for the mRNA that we can use on to do the NGS analysis. And that is of particular interest to us as we continue to move forward because, and this is why we're expanding in this area, is because other people are asking us about other species as well, right? Like, sheep, goat, llama, I get it. We want to work with those as well, and that's why we're developing those kinds of primer sets internally so that we can address those needs to our clients and hopefully for yourself as well. Of course, we're improving our protein sequencing capabilities every day. That is of particular interest to us, and we're actively developing algorithms right now to improve the protein-only sequencing accuracy of our platform. In the projects as they stand, we're delivering a few antibodies out of a given workflow. But of course, we're looking to always expand that, get more antigen-specific leads, express combinatorially different antibodies that might be present in the proteomics data, and deliver more meaningful leads at the tail end of our projects. And of course, looking for new methods of immunizations. What I've shown you thus far is a workflow that works on peptides and soluble proteins, and we're always keen to explore novel immunization protocols in the future as well, stuff like gene guns and DNA immunizations and VLPs and the like. So in summary, I've walked you through the REMAP technology. I've demonstrated that it's a powerful technology that's capable of acquiring the amino acid sequence of any pure monoclonal antibody or protein sample. I've shown that using a combination of proteogenomics, REPAB, can provide a library of antibody sequences that are useful for early-stage R&D, whether that be in therapeutics, diagnostics, or reagent antibody development. And of course, Rapid Novor is seeking royalty-free contracts for use of the technology in the pharma biotech industry. We don't do royalties. We really just do straight fee-for-service at this time. And of course, we're keen to collaborate with anyone else on any other kinds of projects as well, should you already have, other aspects of your pipeline filled in. But of course, we're happy to entertain those collaborations as they come. So again, my name is Anthony. You can reach me at anthony@rapidnovor.com. You can call me on my work phone there as well, or you can just check us out online at rapidnovor.com. If you do ever meet me in this post-pandemic world, you're going to see probably a little more hair than what you see in this picture here. This is my pre-pandemic haircut. I look forward to showing you all what we have going on these days. But yeah, I look forward to hearing from you all. Thanks so much for coming, and I'll I guess leave it to Francis to take us out here.
Great. Thank you, Anthony, for an excellent presentation. We can't wait to see you post-COVID. We've received a few questions already, but we'll give the rest of you a moment to enter your questions in the Q&A box to the left of the slides. I'd like to thank Twitch Bioscience, Gyro's Protein Technologies, Pacifica, Applied Biomath, Rapid Novor, Berkeley Lights, Intertech, and Ajinomoto for sponsoring this Digital Week. Also, be sure to check out the resources list to the right of your screen, where you can download a few featured white papers. Now, back to Anthony to begin the Q&A. So we have a few questions in here. Let me start with the first one. How does the phase 2 screening work?
Yeah, that's a good question. Basically, we will receive a polyclonal antibody sample. The sample should be purified by AG as well as affinity purified against your target of interest. And then, like I said, we're just going to try to sequence it. We're just going to put it through a workflow, see what kinds of data we get, see what kinds of sequences we get, in order to assess, essentially, for lack of a better word, the sequenceability of those samples. Like I said earlier, I think that the real advantage of this approach that we're taking is that we're allowing you as a client to get a lot of shots on goal here. You can try, firstly, different rabbits against the same target of interest, but also you could try and switch up the peptides that you're using as your immunization. And you can assess the sequenceability on that basis as well.
Great. So let's move on to the second question. Can you work with multiple signal peptides and find the best one?
Indeed, yeah. So hopefully this is kind of just, I answered this to some extent in the last reply, but yeah, indeed, we could work with multiple different signal peptides and then find the best one for our purposes, right? This is always kind of the nature of the question, is what is the best one? I'm happy to hear you tell me why an antibody sample is the best one for your application. Of course, when we do the assessment in our hands, we're always assessing the best one with respect to sequenceability. That's not necessarily going to be in congruence with the highest possible affinity, although of course the platform yields high affinity binders because of the inherent affinity purification bias. Yep.
Okay, great. Thank you. The next question is, what types of antigens have you tried to enrich antibody population against? Soluble cells?
Yeah, great question. So we have already done the workflow and enriched for peptides as well as soluble proteins and domains within soluble proteins. So we've, like I said, done a couple of COVID projects. Of particular note, I guess, to this point is that we are trying to find high neutralizing antibody binders against the RBD domain, receptor binding domain of the spike protein, right? So like that, target is the spike protein. Of course, we are trying to choose a particular fraction against just the RBD, and those results are forthcoming. Like those results look pretty good in terms of the mass spec data that we pulled out, in part because we had a really good patient sample that we worked with, highly convergent patient sample that we worked with that works really well for our workflow. It's kind of an interesting point, very anecdotal, but like, we and others have reported that some patients have an extremely convergent antibody response, some don't. We prefer the former compared to the latter, and we have seen that case, and we have run it through the workflow, and the results, like I said, are pretty good. So.
Great, thank you. Next question. Is it possible for RePAB to work through providing affinity purified material and the peptide of interest? This would allow for immunization, and then collected antibodies could be compared back to the affinity purified material.
Sorry, I'm just going to read this one. It's a bit of a long question. Possible to read that?
Sure.
If I understand it correctly, you're asking if we can work with just the affinity purified material and the peptide. This will allow for immunization, I think. Yeah, like, I mean, if you're asking if we can like basically run the platform and compare it against an existing polyclonal antibody, like sure, yeah, we could definitely do that and see what kinds of results we get. This part of like the RePAB workflow is that we do prefer to have a naive sample that we can compare the final bleed to, and as well as like a kind of like mid-schedule sample to compare the final bleed to as well. Affinity maturation, somatic hypermutation, these are interesting phenomena, and of course, like we're still just trying to decode that from our perspective as well. So of course, we could do a comparison and see what kinds of results we get. Yeah.
Okay, great. Next question. Can you work with alloy mice, human IgG transgene mice?
That's a great question. We haven't yet, and we would be keen to try. I should say that like we've been really spitballing this tossing back and forth this question of like working with mice for some time, right? A couple of things. The first problem is like mice are small. Mice are physically very small, and the amount of material that we require to run the RePAB workflow, specifically from the proteomics, often requires like at least 1 milligram of affinity purified IgGs. Like that can be tricky to get out of a mouse for obvious reasons, right? And so for that reason, we've been kind of hesitant to work with them. But it's something that we're keen to explore. We do have some ongoing projects with mice. We're just waiting to get, actually receive those bleeds from our partner and put them through the protocol. But particularly with like these transgenic mice, like I know the advantage. Like you're working with human antibodies, and that's great. And I've got some friends in these companies as well. We've been chatting about it quite a bit. But one of the other problems is actually that a lot of these mice are built for diversity. And that's something that we're keen to explore. I'm not saying it's a deal breaker for us, but it's something that we're keen to check out and observe, because if these mice are built for diversity and they have an extremely divergent response, that can be problematic from the proteomics perspective. What we're keen to try in the very near future is highly inbred strains, I think is where we're kind of sitting on in terms of our thinking about this. So we have highly inbred strains that might produce a more convergent antibody response. This might be better for us with respect to the sequencing workflow, but of course we're happy to talk about it with you and run through the thought experiment before we try anything as well. Yep.
Great, thank you. On to the next question. How much of the purified AB material you use for polyclonal sequencing?
Yep, great question. So hopefully I just answered that one as well. We're looking for roughly 1 mg of the purified polyclonal antibody fraction after the affinity purification. So that's, what we're looking for.
Okay, great. And I think you just answered the next question. So let me go on to the next. In general, do mAbs usually exhibit higher affinities than the parental PAB?
Yeah, that's a good question. We've seen that be the case anyway. The parental PAB, eh? It's an interesting choice of words, but yeah, certainly the case. Like when we did this rabbit immunization against the peptide of interest, like that's what we found, right? So our ELISAs bore that out, that the couple of monoclonal antibodies acting individually outperformed the original polyclonal sera. And I think there's like a couple of different reasons why that might be the case, but like I think the obvious one is like you're working with a pure high affinity monoclonal antibody as opposed to the poly, which could have a lot of like less high affinity antibodies present in it. And so, I think you're getting higher affinity on that basis. The fact that like the antibody itself that we are expressing recombinantly is just a higher affinity binder than the average of that polyclonal sera. Yep.
Great, awesome. So the next question is, rather than purifying the PABs on protein G, should not the immunoaffinity purified on antigen column antibodies be better for LCMS?
Yeah, thanks for the question. The answer is, well, we do both. Yeah, so we do the protein G because we really like to just try to be we want to isolate for the isotype. So we want to just make sure that all the antibodies we're getting are IgGs. We don't look for IgMs or IgAs. We're really just looking to down select to specifically IgGs because this is helpful for just the sheer amount of data that we have to work with. And so yeah, we do the protein G purification, but subsequently we do still do the antigen specific affinity purification as well, which definitely helps when we do the LC-MS workflow. Yep.
All right, great. Next question. From human samples, how many unique sequences you often get? How many mAbs from pub sequencing?
Yep, so from the human sample, it varies a lot, right, between different patients. Like I said earlier, in our COVID case, we found that some people had an extremely diverse antibody response. Some people had an extremely convergent antibody response. At the end of the day, we are looking for a convergent antibody response to put to our workflow because that would be leading to a higher probability of success of us actually pulling out, full-length paired heavy and light chain paired antibodies. So unique sequences, yeah, we get a few different unique sequences that are extremely high affinity and in high abundance in that polyclonal sera. The exact number does vary, but in the projects that we've done so far, it's like between 2 and 5. Yeah.
Okay, great. Thank you. Next question. Have you sequenced any antigens bound to IgM?
Yeah, good question. The answer is no. We typically just do the IgG enrichment, and so we don't usually see too many IgMs in our samples. We'd be keen to talk further with you about that, but yeah, we just haven't done it.
Okay, great. Next question. Are you pulling the most high affinity binders from the sample?
Yeah, great question. And the answer is yeah, we definitely hope so. Indeed, it is the case that we pull high affinity binders using the antigen-specific column. The antibodies that are going to be left after all that washing are going to be just the best ones in terms of their binding affinity. So indeed, yeah, we're pulling high affinity binders out of our purification scheme.
Okay, great. Next question. Are you able to take a PAB without those cells and get the sequences?
Yeah, good question. At this time, no. You really do require that we have the accompanying B cells for the polyclonal antibody sample of interest because we do need to do the B cell repertoire sequencing. We really need the NGS data in order to faithfully recapitulate the full-length sequence before we can, as we're assembling all those different peptides from the poly sera into the most dominant antibodies that we can observe from the proteomics. Yeah.
Okay, great. And then next question, are you able to work with other animals than rabbits?
Animals, no, but human patients, yep. We can certainly work with those samples at this time. And like I said, we're working on it. You know, we're happy to talk about collaborations and potentially explore other other solutions as well. But at this time, yeah, we really just work with rabbits and human patients.
Okay, great. Thanks. Next question. How much antigen needs to be provided for immunization?
Yep, good question. So we look for a few mgs. That exact amount will just depend on like the nature of the antigen, like whether we're talking about like a peptide or a soluble protein or what the case may be. But generally, that's how much we look for. I'm happy to talk about that offline as well. We can definitely, yeah, have a specific conversation about that.
Okay, great. Next question. How to ensure the H and L chains from PAB sequencing are naturally paired?
Yeah, great question. Of course, the pairing of the antibody heavy and light chains is a problem. You know, I've seen other people do this kind of approach that we've outlined here, in which they're expressing, 10 heavy light chains, and then doing like a combinatorial matrix and trying to find the best binders that way. That's not what we're doing. We have a proteomics technique for deriving the how do I say this? Like we've developed internal wet lab techniques on the mass spec to derive the full length sequence, and then pair them. I'm happy to talk a little bit more in detail, perhaps offline. But I guess the easy way to put this is like we're doing a top-down approach. Yeah, like we developed some techniques in-house for faithfully recapitulating that binding activity of the cognate pair. And in fact, we've actually done this not only in the polyclonal antibody sequencing case, but also on occasion for the monoclonal antibody sequencing as well. Where, as I mentioned earlier, in some of the issues with monoclonal antibodies are actually that you'll see multiple chains inside of the sample, whether that be just a light chain or, in some cases, an additional heavy chain as well. You might actually have two full-length antibodies present in a sample of interest at equal concentrations. In which case, in the past, before we had this technique, we'd have to tell people you should express 4 variants. Try each heavy and light chain combo. And then just see which ones are best. But like I said, using our newer techniques, we've been able to faithfully recapitulate those cognate pairs. It worked well in the mono case, and it's worked well in the polyclonal case as well.
Great, thank you. Next question. How many MABs do you get from rabbit from lab material?
Yeah, great question. Like I said, it varies, but like between 2 and 5 is kind of an expectation that we've been able to work with and deliver on thus far. Of course, we would have, this question also comes up sometimes is like, what would you do with the NGS data? One thing that we're keen to explore further is a more robust combinatorial approach. Like once we have those positively identified hits from the proteomics data, go up and down the phylogenetic tree of the NGS and identify very similarly related antibody sequences, and just express those and see if you can deliver some more antibodies from those initial hits. Like I said, we haven't really done that, but like we're keen to explore it, and that might yield better results. But as it stands, we usually sequence 2 to 5 antigen positive antibody sequences from the technique. Yep.
Awesome, terrific. Thank you so much. So that's all the time we have today for questions. Thank you, Anthony, for a great session.